Hello, everyone. My name's Parvinder Hothi. I'm the lab manager at the Ivy Center Research Lab here in the James Tower Swedish Neuroscience Institute. I'd like to thank the organizers for inviting me to present my research today. We have many ongoing projects at the Ivy Center. Today, I'll be talking about one of my main projects, which involves cancer stem-like cells isolated from glioblastoma patients and a high-throughput drug screening platform that we've adopted here at the Ivy Center, and how we've used this methodology to translate our findings into clinical trials for the treatment of glioblastoma. Cancer stem cells are believed to be a subpopulation of tumor-initiating cells found within tumors. They're responsible for tumor growth, therapy resistance, and tumor recurrence. As shown in this figure here, the hallmark characteristics are self-renewal, the ability to proliferate and initiate tumors in vivo. Other common characteristics include the expression of stem cell markers, and the ability to differentiate into the three main CNS cell types: neurons, astrocytes, and oligodendrocytes. One of the challenges with treating glioblastomas, as most people know, is that every glioblastoma tumor is different. Two of the key publications in this area that highlighted this finding are shown here. The reason being there are different subtypes of glioblastomas. They express different gene signatures, and so they respond to treatments differently. The same can be said for the cancer stem cells that are found within these tumors. There are also multiple different subsets of these cancer stem cells found within a given tumor. So this creates the biggest treatment challenge. There may never be one single treatment that will work for every glioblastoma. This has led to a lot of interest in recent years in personalized medicine and a targeted approach, which is looking at tailoring treatments to individual patients. So I mentioned that cancer stem cells are resistant to standard of therapy. So what does this look like in a GBM patient? A GBM patient, once diagnosed, will receive standard of care, surgery, radiation, chemotherapy with temozolomide, the standard of care drug. This reduces the bulk of the tumor, but these cancer stem cells that are resistant will survive, and they will go on to proliferate and generate a secondary, more aggressive tumor. I already mentioned that these cancer stem cells themselves are different subsets within a given tumor. So the question here is, how do we determine which drug should be used to target an individual patient's cancer stem cells? So at the Ivy Center, in order to study this and answer this important question, we started developing patient-derived cancer stem cell lines from the GBM tissue samples. We have a methodology in place in which we mince the tissue, put it through an enzymatic dissociation, put the cells into serum-free media that promote the growth of stem-like cells. These cells can be grown in a number of ways as adherent cultures on monolayer, but the preferred method and what we've actually used in our work at the Ivy Center is growing them in ultra-low attachment dishes as floating spheres known as neurospheres. The cell lines that we generated in the lab, we of course tested that they did have the hallmark characteristics of stem cells. So that involved doing functional assays to see that they could self-renew. They did express stem cell markers. And they had the ability to differentiate into the different cell types. The gold standard being when we took these patient-derived cell lines and injected them into mice, we did have tumor formation. So next, we worked on developing a platform that allowed us to perform cell-based assays with these patient-derived cell lines. So we adopted a high-throughput screening platform in which we add cancer stem cells into the well of 384 well plates. The drugs are added to the wells at different concentrations. The cells and the drugs are co-incubated, after which we add a reagent that allows us to record luminescence, which allows us to quantify ATP, which is proportional to the number of viable cells within the given well. This allows us to identify which drugs are inhibiting the growth of these cancer-like cells. And using this platform, we're actually able to test in the range of hundreds to thousands of compounds in as little as 1 to 2 weeks. It's important to note that when we're doing these drug screens, we're not just working with a drug at 1 or 2 concentration points. We actually generate what's shown here is a dose response curve, which means we have many concentrations of a given drug tested. This allows us to calculate a parameter known as IC50, the half maximum inhibitor concentration, which really measures how much drug is needed to inhibit 50% of these cancer-like cells within vitro. So the lower the IC50, the more potent the drug. And in this manner, we can actually rank and identify potential drug candidates for treatment. So we took our HTS platform and developed it into a phase 01 clinical trial for treating glioblastoma patients with up to 3 drugs identified from a personalized screen using their own cancer stem cells. The primary objective of this trial was to determine feasibility of the HTS approach. Can we do this in a clinically relevant timeline, and is it safe for the patients to receive these 3 random drugs identified from our HTS screens? In the phase 0 part of the trial, a patient comes in with newly diagnosed GBM. We collect consent, surgery is performed, we obtain the cancer stem cells, we grow these up in the lab over a couple of weeks. We then test the cancer stem cells against a panel of 76 FDA-approved drugs. The drugs on this panel were selected from previous bench studies in which we tested thousands of chemical libraries to identify a subset of drugs that showed high efficacy against patient-derived stem cell lines. Once we have completed IC50 analysis of these dose response curves that we generate, we perform a drug-drug interaction review after selecting the 3 compounds to determine if there are any overlapping toxicities. Once the drug therapy has been reviewed and approved by the Data Safety Monitoring Board, it was being administered to patients in the phase 1 part of the trial once they showed signs of disease progression. So this wasn't the ultimate study design. It was not ideal by any means because obviously we're taking tumor at the newly diagnosed stage, testing the stem cells derived from the first tumor, but we weren't initiating treatment until patients had recurred. The reason being we had to design the study this way was because that was the regulatory guidance we had trying to get this approved by the FDA, with it being the first of its kind trial at the time. We were advised to start with recurrent GBM patients just to obtain the safety data before we could move to the newly diagnosed setting. I mentioned that the drug panel was based on previous bench studies that we'd done to isolate drugs of interest. The panel itself is made up with compounds from many different drug classes, different mechanisms of actions, lots of anti-neoplastics that are commonly used in cancer therapy, but also a lot of unusual FDA-approved drugs that are used for other indications, but not commonly used for cancer. For example, anti-inflammatories, antihypertensive agents, antipsychotic agents that we were seeing were somewhat inhibitory against the growth of these cancer stem-like cells derived from GBM patients. Here is an example of the data that we generated during this first clinical trial. So you'll see here in panel A, a dose-response curve for 4 different GBM patients against a given drug with some varying degrees in IC50. And then, interestingly, here you'll see in panel B that we do have, with some drugs and some patient-derived cell lines, these huge differences in which we have some patients that do not respond to a given drug at all, but then we have some patients that are clear responders. And by doing this high-throughput screening in this manner, we're able to identify drugs and then rank them in order of potency to basically create a personalized cancer stem cell drug profile that the treating provider can then use to select 3 drugs for then treating the patients when they become eligible to receive treatment. So in addition to looking at just the IC50, we also are taking into consideration other factors when selecting which drug we should use to potentially treat the patient. That includes Lipinski's rule of 5, which are basically parameters around what a good drug should be. An orally active drug, for example, does not have a log P greater than 5. It usually has a molecular mass that is not greater than 500 Daltons, no more than 5 hydrogen bond donors, no more than 10 hydrogen bond acceptors, and the number of rotatable bonds should be less than 10. Importantly, for drugs treating brain tumors, we also want to look at the ability of the drug to penetrate the blood-brain barrier. And for that, we take into consideration the polar surface area of the molecule. PSA is the sum of the polar atoms. The lower the PSA, the better the BBB penetration. Typically, drugs that have a PSA of less than 120 tend to cross the blood-brain barrier, and those that are below 90 are fairly readily absorbed. And then we also look at the brain-to-plasma concentration ratio. So an example is shown here for temozolomide, the standard of care drug. Molecular mass less than 500. It has no violations of the rule of 5. It has a low polar surface area. These are the results from the phase 1 of the recurrent HTS study. So again, the objective here was feasibility and safety of our cancer stem cell HTS approach. We were hoping to treat 10 patients. We managed to treat 9. There were logistical issues, and so we could not get the 10th patient on study, but this study is now considered complete, and we are currently in the process of writing up a manuscript on our findings. The key takeaway here is that each of the 9 patients treated received a unique 3-drug combination. There were no study-related serious adverse events and no unexpected adverse events from the investigational drugs. The median overall survival for GBM is typically 15 months. In this study, the median overall survival for the patients that were treated successfully on study was close to 21 months. So although it sounds like a relatively small study in which we wanted to treat 10 patients, it actually was a technically very challenging trial. We completed the phase 0 part of the study, which was generating the cell lines and performing the screens on 45 GBM patients. So we did manage to create a very rich data set of drug sensitivity profiles. So we used this data in a collaboration with the Institute for Systems Biology to generate an R01, which is currently ongoing. And in the R01, we're trying to understand the drug resistance that we see and identify potential synergistic combinations for the treatment of patients. We currently have a manuscript under review that's looking at the mechanisms of drug resistance in these patient-derived stem-like cells. So now we have received approval from the FDA and IRB to launch a new HTS study in which we are using the same approach, but this time upfront for treating newly diagnosed glioblastoma patients. The primary objective of this study will be safety and potential efficacy of the investigational treatment, 3 HTS drugs determined from the cancer stem cell screens, alongside standard of care. In the treatment phase of the study, 10 patients will receive their unique 3-drug combination alongside radiation and TMZ. The protocol also allows patients to use the Optune device should they and their treating provider agree this to be safe. The main difference between this study and our recurrent study is that patients do not have to wait for tumor recurrence to receive their personalized therapy. So we're very excited to be able to launch this trial for newly diagnosed GBM patients, and we expect to open for enrollment next month. So the advantages of the new study is that we have a well-established methodology and pipeline in place. Our team has a proven track record in cancer stem cell isolation, HTS assays, and we have the ability to perform drug-drug interaction reviews that give us good safety outcomes. Because we generated so much data from the first study, we've been able to refine the GBM drug panel for the newly diagnosed study. We expect there to be a lot less challenges in this trial because patients are receiving treatment shortly after tumor resection, at a time when tumor burden is low and they receive their personalized therapy prior to disease progression. Another interesting point from this new study is that it will give us the ability to isolate cancer stem cells from a patient's primary tumor and recurrent tumor, giving us a matched pair for analysis. I was able to do this for one patient. As shown here in the blue dots, you see a dose response curve for the recurrent tumor. In comparison, when tested the stem cell line from the primary tumor from the same patient, the response to the drug was much greater. This suggests that giving patients a personalized therapy upfront as close to tumor resection as possible will have greater therapeutic potential. I'd like to thank the Ivy Center team, Dr. Charles Cobbs and Dr. Alipi Bonham, who are both also co-investigators on the new HTS study, the lab team, Ralph, Lee, and Yoon, Nathan Hansen, our wonderful research coordinator, and the neuroscience research director, Danielle O'Connor. I'd also like to thank the Swedish Foundation, our wonderful partners who do fundraising to support our work at the Ivy Center. And I'd also like to thank you all for listening.